It is really wonderful how nature has devised the machinery of life. Man has always tried to explore the mysteries of nature. In this pursuit of gaining knowledge, man has been able to crack some of the codes of Mother Nature. DNA and RNA are such two important codes. After DNA and RNA were discovered and their structures were well studied, it was important to know how actually they function in harmony. DNA, as we know, is the set of genetic instructions governing cellular activities. These instructions are implemented via the synthesis of RNAs and proteins.
Hello students, I welcome you to today's topic on transcription. DNA, RNA and proteins are three important elements that you must know before studying this topic. ਜਿਵੇਂ ਕਿ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ DNA ਵਿੱਚ ਕੰਪਲੀਟ ਜੈਨੇਟਿਕ ਇਨਫੋਰਮੇਸ਼ਨ ਹੁੰਦੀ ਹੈ ਜੋ ਕਿ ਆਰਗੈਨਿਜ਼ਮ ਦੇ ਸਟਰਕਚਰ ਅਤੇ ਫੰਕਸ਼ਨ ਨੂੰ ਨਿਰਧਾਰਿਤ ਕਰਦੀ ਹੈ। DNA ਤੋਂ RNA ਬਣਦਾ ਹੈ ਅਤੇ RNA ਤੋਂ ਪ੍ਰੋਟੀਨ ਬਣਦੇ ਹਨ। RNA is an intermediary stage between DNA and the protein making factories, the ribosomes. RNA serves to carry information from the genome to the ribosomes. Proteins could be viewed as the programs of the cell. They are the physical representation of the information contained within the genome. So we can say that DNA is long-term storage. It is stable, packaged and inert. RNA is a short-term storage. It is unstable and lacks secondary structure. Some RNAs have enzymatic activity. Proteins are the programs of the cells. They are the physical manifestations of the abstract information recorded in the genome. ਕਿਉਂਕਿ ਅੱਜ ਅਸੀਂ DNA ਅਤੇ RNA ਨਾਲ ਰਿਲੇਟਡ ਟੌਪਿਕ ਬਾਰੇ ਪੜ੍ਹ ਰਹੇ ਹਾਂ ਜਿਸ ਵਿੱਚ ਟਰਮ ਸੈਂਟਰਲ ਡੌਗਮਾ ਨੂੰ ਕਈ ਵਾਰ ਇਸਤੇਮਾਲ ਕਰਨਾ ਪਏਗਾ। ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਸੈਂਟਰਲ ਡੌਗਮਾ ਬਾਰੇ ਯਾਦ ਹੋਵੇਗਾ। ਜੇ ਨਹੀਂ ਤਾਂ ਆਓ ਛੇਤੀ ਨਾਲ ਇਸ ਨੂੰ ਰਿਕਾਲ ਕਰ ਲਈਏ।
Central dogma. Central dogma forms the backbone of molecular biology. In this, the DNA forms new DNA strands through replication. The DNA codes for the production of messenger RNA through the process transcription. There is another process known as translation, where RNA sequence is translated into a sequence of amino acids, resulting in the formation of a protein.
In eukaryotic cells, the second step, transcription, is very important because it is not possible to translate DNA directly into protein. So students, today we will focus on transcription. Before I elaborate on this topic, let us see the learning objectives for today's lesson.
Upon completion of this topic, learners will be able to define transcription, discuss the need of regulation of transcription, describe the process of transcription, differentiate between prokaryotic and eukaryotic transcription.
Let us start with the topic. The main goal of transcription is to make a RNA copy of a gene. The process by which information stored in DNA is converted into RNA is called transcription. In transcription, an RNA polymer is made from a DNA template. This RNA is either used as it is or delivers its information to an
organelle called a ribosome.
These ribosomes are protein-making factories. Ribosomes take RNA-coded information and translate that information into strings of amino acids called polypeptides by the process translation. This RNA can direct the formation of a protein. In normal cells, the transcription process is very tightly regulated. There are a few important things to be remembered. Genes must be transcribed at the correct time. The RNA produced from a gene must be made in the correct amount. Only the required genes should be transcribed. Turning transcription off is just as important as turning it on. ਤੁਸੀਂ ਇਸ ਨੂੰ ਕਿਸੇ ਫੈਕਟਰੀ ਦੀ ਸੋਫਿਸਟੀਕੇਟਡ ਪ੍ਰੋਡਕਸ਼ਨ ਲਾਈਨ ਦੇ ਨਾਲ ਕੰਪੇਅਰ ਕਰ ਸਕਦੇ ਹੋ। ਜਿਵੇਂ ਤੁਸੀਂ ਫੈਕਟਰੀ ਵਿੱਚ ਵੇਖਦੇ ਹੋ ਕਿ ਜਦੋਂ ਕਿਸੇ ਪ੍ਰੋਡਕਟ ਦੀ ਜ਼ਰੂਰਤ ਹੁੰਦੀ ਹੈ ਤਾਂ ਲੋੜ ਅਨੁਸਾਰ ਪ੍ਰੋਡਕਸ਼ਨ ਵਧਾ ਦਿੱਤੀ ਜਾਂਦੀ ਹੈ ਤੇ ਜਦੋਂ ਪ੍ਰੋਡਕਟ ਦੀ ਜ਼ਰੂਰਤ ਨਾ ਹੋਵੇ ਤਾਂ ਪ੍ਰੋਡਕਸ਼ਨ ਬੰਦ ਕਰ ਦਿੱਤੀ ਜਾਂਦੀ ਹੈ। Now, let's discuss constituents of a transcription unit. To explain this point, a hypothetical sequence from a transcription unit will be shown here.
Constituents of transcription unit. The stretch of DNA that is transcribed into an RNA molecule is called a transcription unit. So, transcription unit can be defined as the segment of DNA that takes part in transcription. A transcription unit is a linear sequence of DNA that extends from a transcription start site to a transcription stop site. A DNA transcription unit that is translated into protein contains sequences that direct and regulate protein synthesis in addition to code the sequence that is translated into protein. A transcription unit in DNA is defined primarily by the three regions in the DNA: a promoter, the structural gene, and a terminator. This is a transcription unit. The promoter and terminator flank the structural gene. The promoter is nothing but a DNA sequence that lies upstream of the RNA coding region to the 5' end. The promoter is not actually transcribed. Its role is purely regulatory. It provides a binding site for RNA polymerase. In many cases, the promoter has an AT-rich region called Tata box. Terminator region is present downstream at the 3' end of the coding strand. Coding and non-coding strands. As you all know, DNA is made up of two strands. Since two strands have opposite polarity, the strand that has the polarity 3' to 5' acts as a template and is also referred to as template strand or non-coding strand. The other strand, which has the polarity 5' to 3', does not code for anything, is referred to as coding strand.
Let's discuss the starting enzyme required for transcription. Apart from some initiation factors, RNA polymerase is an enzyme which is required for initiation of the process. RNA polymerase helps in unwinding the DNA strands. So, we can say that RNA polymerase is needed for constructing RNA chains from DNA during transcription. You know, RNA polymerase enzymes are essential for life and are found in all organisms and even in many viruses. There are at least 3 RNA polymerases. There is a clear-cut division of labor. I hope you all know very well that there are 3 types of RNAs: transfer RNA, messenger RNA, and ribosomal RNA. The RNA polymerase 1, also known as polymerase 1, is found in nucleus and transcribes
ribosomal RNA.
Whereas the RNA polymerase 2, also known as polymerase 2, found in nucleoplasm, is responsible for transcription of messenger RNA. The RNA polymerase 3, also known as polymerase 3, is also found in nucleoplasm, transcribes precursor of transfer RNA. Polymerase 1 transcribes rRNA. Polymerase 2 transcribes mRNA. Polymerase 3 transcribes tRNA. After this, let us now go through the various stages of transcription. Transcription is divided into 4 stages: initiation, promoter clearance, elongation, termination. Now, let's study these stages one by one.
Initiation. RNA polymerase binds itself to the promoter region. 2 strands of DNA uncoil progressively from the site of polymerase binding. Sigma factor recognizes the start signal. In most prokaryotes, chain opening occurs in the region occupied by TA, TAA, TG nucleotides. In case of eukaryotes, the polymerases do not directly recognize their core promoter sequences. Here, a collection of proteins called transcription factors mediate the binding of RNA polymerase and the initiation of transcription. The RNA polymerase proceeds to read one strand moving in its 3' to 5' direction. Ribonucleoside triphosphates present in the surrounding medium come to lie opposite to the nitrogen bases of the DNA template. Ribonucleotides containing 3 phosphate groups hydrogen bond through the process of complementary base pairing with the exposed deoxyribonucleotides on the unwound strand. They form complementary base pairs: uracil opposite adenine, adenine opposite thymine, cytosine opposite guanine, and guanine opposite cytosine. Synthesis of the RNA proceeds in the 5' to 3' direction. The ribonucleotides are then covalently bonded together by phosphodiester bonds. A pyrophosphate is released from each ribonucleoside triphosphate to form ribonucleotide. RNA polymerase moves along the DNA template, causing elongation of RNA chain at the rate of some 30 nucleotides per second. This phase is known as elongation. RNA synthesis stops as soon as polymerase reaches the terminal region. Rho factor is required for termination. Rho factor destabilizes the interaction between the template and the mRNA, thus releasing the newly synthesized mRNA from the elongation complex. The released RNA is called primary transcript.
Termination of transcription. There are a number of proteins involved in termination of transcription, particularly this protein called Rho. However, the mechanism by which Rho terminates transcription is not understood. This released RNA needs to be processed to form functional RNA. So here we have seen that one strand of DNA, the template strand, or we may call it as non-coding strand, was used as a template for RNA synthesis. As transcription proceeded, RNA polymerase traversed the template strand and used base pairs complementary with the DNA template to create an RNA copy. Although RNA polymerase traversed the template strand from 3' to 5'. The coding strand is usually used as the reference point. So, transcription is said to go from 5' to 3'. DNA replication ਦੇ ਉਲਟ mRNA transcription ਵਿੱਚ ਬਹੁਤ ਸਾਰੇ RNA polymerases involve ਹੋ ਸਕਦੇ ਹਨ ਕਿਉਂਕਿ multiple rounds of transcription may take place, meaning many mRNA molecules can be produced from a single copy of a gene. The last step is termination. After termination, as I told you, the released RNA needs to be processed to form functional RNA. All these changes come under the heading of post-transcription processing. In fact, primary transcript is often larger than the functional RNAs. Let's discuss post-transcription processing, which is required to convert primary transcript of RNAs into functional RNAs.
Post-transcription processing. If we see eukaryotic transcripts, they possess extra segments called introns as intervening sequence or non-coding sequence. The functional coding sequence are called exons. Capping and tailing. Capping involves a modified guanine which is attached to the 5' end of the pre-mRNA. This cap protects the RNA from being degraded by enzymes that may degrade RNA from the 5' end. Tailing involves the synthesis of the poly-A tail. When a special poly-A attachment site in the pre-mRNA emerges from the RNA polymerases 2, the transcript is cut there, and the poly-A tail is attached to the exposed 3' end. This step is also known as polyadenylation. Splicing is the removal of introns and fusions of exons to form functional RNAs. There is a step-by-step removal of introns present in the pre-mRNA and splicing of the remaining exons. Each intron is recognized by components of splicing apparatus of snRNPs. A complex called spliceosome is formed between 5' end and 3' end of intron. With the removal of introns, the adjacent exons are brought together. The ends are sealed by RNA ligase.
Prokaryotes ਵਿੱਚ mRNA ਨੂੰ ਐਕਟਿਵ ਹੋਣ ਲਈ ਕਿਸੇ ਲੰਬੀ ਚੌੜੀ ਪ੍ਰੋਸੈਸਿੰਗ ਦੀ ਲੋੜ ਨਹੀਂ ਹੁੰਦੀ। ਹੁਣ ਅਸੀਂ ਪ੍ਰੋਸੈਸ ਆਫ ਟ੍ਰਾਂਸਕ੍ਰਿਪਸ਼ਨ ਬਾਰੇ ਜਾਣ ਗਏ ਹਾਂ। ਆਓ ਹੁਣ ਪ੍ਰੋਕੈਰੀਓਟਿਕ ਅਤੇ ਯੂਕੈਰੀਓਟਿਕ ਟ੍ਰਾਂਸਕ੍ਰਿਪਸ਼ਨ ਵਿੱਚ ਡਿਫਰੈਂਸਸ ਵੇਖੀਏ।
Differences between prokaryotic and eukaryotic transcription. Prokaryotic transcription occurs in the cytoplasm, whereas eukaryotic transcription occurs inside the nucleus. Transcribed products move out into cytoplasm for translation. In prokaryotes, transcription is coupled with translation, whereas in eukaryotes, transcription and translation are separate processes. Prokaryotes have only one RNA polymerase, which synthesizes all types of RNAs, whereas eukaryotes have 3 RNA polymerase. Polymerase 1 for ribosomal RNA, polymerase 2 for messenger RNA, and polymerase 3 for transfer RNA. Messenger RNA formed in prokaryotes is polycistronic, whereas eukaryotic structural gene is monocistronic. In prokaryotes, mRNA is not modified. Eukaryotic mRNA is modified through RNA splicing, capping, and the addition of a polyadenylation tail.
With these differences, we come to the end of our main topic. Let us quickly summarize what we have learned today. The genetic information that is passed on from parent to offspring is carried by DNA of a cell. In order for DNA to produce the proteins, it must first be transcribed to RNA through a process known as transcription. Thus, transcription is defined as the transfer of genetic information from DNA to the RNA. There are 3 different phases involved: initiation, elongation, and termination. To initiate the process of information transfer, one of the strands of the double-stranded DNA serves as a template for the synthesis of a single strand of RNA, that is, complementary to the DNA strand. The enzyme RNA polymerase binds to a particular region of DNA that is termed as promoter. Once the enzyme is bound to the promoter, it unwinds the DNA and starts to make a strand of RNA with a base sequence complementary to the DNA template. Once transcription is initiated, the process of elongation starts. The substrate for RNA polymerase are nucleoside triphosphates. The next phase is called termination. There are number of proteins involved in termination of transcription, particularly a protein called Rho. Once this termination signal is recognized by the RNA polymerase, it releases the DNA and transcription ceases. The newly synthesized RNA strand, which is also known as primary transcript, now undergoes post-transcriptional processing. A newly synthesized RNA strand consists of both coding sequences, exons, and non-coding sequences, introns. This primary transcript that is formed at the end of transcription actually passes through post-transcriptional changes. Capping and tailing takes place and a process called RNA splicing occurs and the introns are removed. The remaining exons are joined together to form the final mRNA product, which codes for a single protein. So students, enough for today. Let's quickly check with some short questions what you have grabbed today. Be attentive. Here are some questions. Question 1. Define central dogma. Answer is, the central dogma forms the backbone of the molecular biology. There are 3 stages in it. DNA is replicated to DNA through the process replication. Then DNA is transcribed to RNA, which is further translated to protein. Protein is never translated back to RNA or DNA except for retroviruses. The central dogma is replication, transcription, translation. Next question. The process by which DNA makes RNA is called? The answer is, the process by which DNA makes RNA is called transcription. And my next question is, a promoter gene defines the site for initial binding of RNA polymerase, direction of transcription, copying the strand, all of the above. And the answer is, a promoter gene defines the site for initial binding of RNA polymerase. My next question is, how will you define transcription? And the answer is, the process by which stored information in DNA is converted into RNA is known as transcription. Next question. Define transcription unit. And the answer is, the segment of DNA that takes part in transcription is called as transcription unit. Another question. How many types of RNA polymerase are there in eukaryotes? And the answer is, there are at least 3 RNA polymerases in eukaryotes. Polymerase 1 transcribes rRNA, polymerase 2 transcribes mRNA, polymerase 3 transcribes tRNA. My next question, in which direction does transcription takes place? And the answer is, transcription takes place in 5' to 3' direction. Next question, in which direction does the RNA polymerase read? Answer is, RNA polymerase reads in 3' to 5' direction. Next question. Name the 3 regions of this transcription unit in DNA. Answer. A transcription unit in DNA is defined primarily by 3 regions. These are a promoter, the structural gene, a terminator. Next question. In dash transcription and translation take place side by side. Answer is, in prokaryotes, transcription and translation take place side by side. Question. Removal of introns in a transcript RNA is known as dash. And the answer is, the removal of introns in a transcript RNA is known as splicing. My next question. What is the major difference between prokaryotic and eukaryotic transcription? The answer is, prokaryotic transcription occurs in the cytoplasm alongside translation, while eukaryotic transcription is localized to the nucleus. Second major difference is regarding modification of transcript RNA. In prokaryotes, mRNA is not modified. Eukaryotic mRNA is modified through RNA splicing, capping, and the addition of a poly A tail. Hope I have made a successful attempt to clear the concepts regarding the process of transcription. Thanks for your cooperation and attention. Looking forward to the next class. See you then.